The broadcast is now starting. All attendees are in listen-only mode.
Good evening. This is Daniel Roth with Brushy Mountain Bee Farm, and I'm very excited about today's topic. Just to let everybody know, this is a recorded event, and we will be uploading it to YouTube upon completion. So you can come back, you can watch this again to better understand what's going on. It is a recorded session. At the end of the discussion here, we will have a question and answer period. So please hold on to those questions until then. And again, like I said, this is recorded, so you can come back in and watch this. Brushy Mountain Bee Farm has been doing a lot of product development over the course of some years. We're trying to help encourage propolis production and ultimately increase colony health. During the research stage of this product development, we turned to the leading experts in the industry, and our guest panelist today, Marla Spivak, is the source when it comes to understanding propolis. Marla's research and focus on propolis through the Bee Lab at the University of Minnesota is helping beekeepers like me, like you, understand the health and the immunity system of honeybees. When I first started out as beekeeping, I was excited but clueless. You know, I knew bees were instrumental in agricultural, but it wasn't until I heard a TED Talk by Marla that I fully grasped their importance. And I'm sure everybody's heard it, or at least seen the pictures where, you know, you see a supermarket with all the fruits, the vegetables there, and then another image that has all those fruits and vegetables missing. You know, it was really that TED Talk that really brought me in to the light of Marla and what she was advocating for our honeybees. She is a MacArthur Fellow and McKnight Distinguished Professor in Entomology at the University of Minnesota. She has been distinguished for many other awards. She helped develop the Minnesota Hygienic line of queens. Ultimately, all around, she's a wonderful person who is passionate about bees. Please listen in as Marla enlightens us on propolis and the health of our colony. So Marla, please continue on this conversation.
All right, thank you, and hello everybody, good evening. It's Minnesota summer, it's beautiful up there, and I hope it's not too hot wherever you all are. So I'm going to give a talk on propolis. You can pronounce it propolis or propolis, it doesn't matter, but we're going to talk about why you should care about it. So most of you are very familiar with propolis because it's sticky and it makes it very difficult to manage a colony of bees, to pry the boxes apart, to get the frames out of the boxes. If you get it on your clothes, it's there forever. But it's actually really good stuff, and I will explain to you why. I'll be showing you some data slides and then other picture slides, but I hope this all makes sense. So what is propolis? Propolis is actually plant resin. So it comes from plants, and sometimes when bees collect it, they mix it with a little bit of beeswax. But for the most part, it's just straight plant resins. So what does that mean? So plants, when they're stressed or diseased, they produce resin. And they produce this resin in different parts of the plant. They can produce it from the bark, they can produce it from the leaf buds, or some in the tropics, some plants secrete it from the flowers. But the resins that come from these plants are highly, highly antimicrobial, meaning they're antibacterial, they're antifungal, they're antiviral, and they're full of terpenes and flavonoids and compounds that are have a lot of health benefits. So the bees find these plants that are producing these resins, and they scrape the resins off the plant. In this case, what you're looking at is a balsam poplar leaf, and the red on the little new leaves is the resin. So the bees go and scrape that resin off those new leaf buds, and they pack the resin on their hind legs. So in the lower left photo is this bee with bright red resin, and in the right-hand photo is a bee in the tropics in Brazil, and she's collecting resin from a shrub The scientific name is Baccharis, and the resin from that plant is green, and you can see the green resin on her back legs. So they put the resins where they would put pollen if they were collecting it from a flower. And then they bring the resin back to their nest, wherever they're nesting. And honeybees in the wild, they nest in hollowed out tree snags, old tree snags. And what we're showing on the right is an old tree that's been cut in half, and all the beeswax combs were removed, or most of them, and you can see the remnants. And then you can see this darkly stained area that was surrounding the combs. So within the tree cavity, the bees deposit these resins as a lining or a coating within their tree cavity on the walls of the nest. And we call that a propolis envelope. But in a modern day beekeeping equipment, that's not where they put it, of course. So when they come back, and I'll get back to the modern day beekeeping equipment. So when a bee comes back with that resin on her hind legs, she can't get the resin off her own legs. It's way too sticky. So in this photo, you see a bee removing the resin for another bee, one of her sisters removing that resin from her hind leg. And then together, they kind of cement that resin onto the walls or the cracks and crevices of the nest. And once it's in the beehive, we call it propolis. So actually resin and propolis are the same thing, only in the beehive, they may add a little bit of beeswax to the resin. So that's what propolis is. It's just plant resins. And we don't think the bees are adding any chemical secretions or any other thing to change the chemical composition of the resins. They're just adding sometimes a little bit of wax. So these are photos of that bee tree that we collected to look at the propolis envelope, and you see the entrance on the upper left and upper right. And then the bottom photo is it's a picture from the bees' perspective looking out their front door. And this is within a tree cavity, and you can see all that orange and yellow and brown and green. That's all propolis. And toward the entrance in particular, there's a very, very thick coating of it, sometimes, you know, quarter, half-inch thick. So they really use a lot of these resins inside, or propolis inside. So you're going to hear me interchange the word resin and propolis sometime, and that's because they're really the same thing. Just as beekeepers, we call it propolis. Okay, so why do they collect these resins, or why do they bring in this propolis stuff anyway? It's really energetically demanding for them. Like I said, they can't get it off their own hind legs. They're not eating it, so there's no individual food reward. So on the left, you see a bee with resin on her hind legs, and on the right, a bee with pollen. But the bees are not eating these resins. They're using them for a reason, but they're not using them as food. So why would they collect this stuff? So over the years, we've been addressing these general questions, and the people in the bottom, Mike Simon Finstrom, Renata Borba, and Mike Wilson, were all graduate students in my lab, and they now all completed their PhD studies answering one or several of these questions. So what is the benefit of having that propolis envelope inside the beehive, the bee nest? And if colonies have this propolis envelope, are they healthier? Do they have less disease? And when bees are foraging for these resins outside in the environment, are they choosing them? Choosing different resins? If they're sick, are they collecting more resin? In other words, are they medicating themselves? Are they self-medicating? And then where are they getting these resins from? And are some resins better for them than others? So a whole list of questions that we've been studying over the years. So to begin to answer some of these questions, here's Mike Simon Finstrom. He was the first graduate student, and he's now down in the Baton Rouge USDA Bee Lab. And his question was, does this propolis envelope benefit the immune system of honeybees? So for these experiments, the colonies were healthy. I mean, we had treated them for mites, and as far as we could tell, they had no other symptoms of diseases. And what he did was take propolis we just collected from colonies, and we made an extract. In other words, we dissolved the propolis in ethanol, and we made an extract that we varnished or basically painted inside small boxes. So you're looking at some 5-frame nuke boxes, and we painted the inside either with propolis from Minnesota or propolis from Brazil, that green propolis. And then we put bees and combs and everything in these boxes, and the one on the left is a control. In other words, it just had the ethanol, the alcohol painted on it, and it evaporated in case that had activity. We put the bees in there, and we marked baby bees, and we collected them when they were 7 days old. And then we take them into the laboratory where we look at, this gets a little technical, but we look at transcription levels of antimicrobial peptides. And what that means is antimicrobial peptides are part of the immune system that fights off microbes. And transcription levels is just how much of them they're being produced. And so what you're seeing, 2 of the peptides, hymenoptacin, and another, actually not a peptide, but another part of the immune system called am-eater, which are strange names, of 7-day-old bees. If you look at the levels relative to controls, the levels are lower. So I'll use my pointer. So the levels here are lower than the blue bar, the controls. Brazilian propolis is lower. And these, even though the graph looks upside down, the bars go lower than the controls. And what this means is their immune systems are producing less immune responses. And at first it seems kind of strange. Why would that be? I'm sorry, I went too far ahead. It indicates that when there's propolis, and a lot of propolis in the nest, that whole propolis envelope, that the bees are not having to invest as much energy into their immune systems as they are when there is no propolis, as they have to when there's no propolis envelope. So let me go on and then I'll give you an analogy, okay? So this is Renata's experiment. And she wanted to know if that benefit to the immune system persists throughout the year. So Mike Simon Finstrom's experiments were done just in 7-day exposure to bees within that propolis envelope that he painted on. And Renata wanted to see if that benefit persisted through the year. And so she used slightly different methods. She took propolis traps. These are commercial propolis traps. They're plastic sheets that basically you put on top of the top bars of a colony. But in this case, we cut them and stapled them inside the brood chambers of our brood boxes. And you can see when we removed them, the bees had stuck a bunch of propolis to the walls through those little slats. And that's the propolis envelope. We actually left the propolis traps in the colonies for the whole year. We didn't remove them like the photo. I just wanted to show what it looked like. And we put, for 2 different years, 2 whole new sets of colonies, a bunch of bees in these boxes with the propolis traps and without the propolis traps. And then we measured their immune systems through time and their health through time. And I'll just show a little of that data. And again, when they had propolis, in this time it's in these maroon bars, the expression of all of these antimicrobial peptides and functions of the immune system were always lower than bees from colonies without a propolis envelope. So what this means is, in both of those experiments, that bees do not have to invest as much energy in their immune response, probably because the propolis was killing off many of the microbes in the nest cavity. So the analogy that may help you understand this is if I lived in a house with a lot of mold, and I had an allergic response to that, I'd be sneezing, my immune system would be elevated, and if you measured my immune system like we did to the bees, you'd see that those bars would be high, were very, very high. But if I painted all the walls in my house with propolis, or smeared it all on the walls of my house, it would kill off the mold, and then my immune system would be quieter again. So if we measured my immune system with the propolis in the walls, it would be very, very quiet and lower expression than what we see if there was no propolis. And I hope that makes some sense to you. So this in general means that the propolis is really helping the immune systems of bees, keeping the immune system running at a good energy efficient way. So the next set of questions we asked was, well, that was on healthy bees. What happens if we have colonies with a propolis envelope and then we challenge them? We make the colonies sick. So does the propolis envelope have a therapeutic effect or against pathogen, against diseases? So we started by, again, with Mike Simon Finstrom's experiment, and where he painted the propolis envelope in the boxes, and then we put frames and bees in these boxes. And then we fed the colonies pollen patties that had chalk brood mummies in them. So chalk brood is a fungal disease, and the name is Ascosphaera apis. That's its scientific name. We collected chalk brood mummies from colonies. You homogenize them, you ground them all up, and you mix them with pollen patty, and you feed them to the bees. And within a week or two, they get sick. And so we found that colonies that had the propolis envelope like this had, on average, almost 15 mummies. But if they didn't have the propolis envelope, on average, they had 108 mummies. So this shows us that the propolis was really reducing the amount of chalk brood in the colonies. It was helping the bees. It didn't get rid of it, but it was really lowering the amount of disease in the nest. And Renata then came along and then said, following Mike, and said, okay, so if it works for chalk brood, the fungal disease, does it also work to lower the amount of American foul brood, which is a bacterial pathogen, and it's caused by, the scientific name is Penibacillus larvae. So we call it P larva. And this is what it looks like. I hope you never see it. It's very stinky. It causes the brood to turn into this brown, very stinky, gooey, decomposed mass. And the larvae get it when adult bees are feeding the larvae. If there's any of the spores around, the adult bees inadvertently put some of the spores in the food of the young larvae, and as they grow, they get sick and then they die. So what Renata did in her case, remember she used the propolis traps stapled inside the colonies like this. She had some colonies with propolis traps and some without propolis traps. And then some of them she challenged with the bacteria that causes American foul brood. And the way you do that, she made a spore solution in sugar syrup here and sprayed it on each comb, each side of the comb. And then you wait a little while and the colonies come down with American foul brood. So today for this, just to help make the point clear, I'll talk about colonies that had a propolis envelope and colonies that did not have a propolis envelope, and the ones that we challenged or gave the American foul brood bacteria to. And the hypothesis was that, our idea was that colonies with the propolis envelope and that were challenged with this bacteria would have less American foul brood than colonies without the propolis envelope, and that the larval food, the food that the bees were giving to the larvae, would have greater antimicrobial activity. And I'll explain a little bit of that when I show you the results, why we thought that. So the first thing we found was that, again, if you follow these maroon colored bars, colonies that had the propolis, excuse me, had much less American foul brood compared to colonies that did not have the propolis envelope. And this is a severity score. 0 is 0. If you have a score of 1 of American foul brood, that's just a few cells containing the disease on a frame. A 2 is like you're getting up to 20 cells, disease cells per frame, and 3 is you're just loaded with American foul brood. So you can see that even when they were got some disease, the disease level was much lower than it was than colonies without the propolis envelope. So it was really helping them reduce their disease load. And then what Renata did was, excuse me, one of the ways that honeybees can defend themselves against bacterial pathogens is the adult bees, when they're feeding, put antimicrobial substances in the food. They're just in there. And so Renata took these tiny little larvae out of the cell and scooped up the food and put it in in an experiment where she put it in these little well plates and put a medium in there, and then put a little bit of bacteria from American foul brood in these little well plates, and quantified, measured how much bacterial growth you could see when there's the food in there. So it's kind of complicated, but it's fascinating, because here you show that the food from colonies that had the propolis envelope did not support the growth of the bacteria compared to brood food from colonies without the propolis envelope. So I'll say that again. This is brood food from colonies with a propolis envelope, and it shows how much bacterial growth they supported in here, and it's much less than the food from colonies that did not have that propolis envelope. So we have no idea how this is working. We don't know if the nurse bees are making more of those antimicrobial peptides and putting them into the larval food, or maybe it's the volatile compounds from the propolis, just it's in the air of the colony, and those volatile compounds just move into the larval food and make it more antimicrobial. We don't know the answer to this, and these are some questions we're trying to explore this summer in the laboratory. So now we know that the propolis is really good for the immune systems of bees when they're healthy. It keeps their immune systems at an efficient expression level. And we know that when bees are sick, the propolis envelope helps reduce the disease load. And so now we started to ask some really crazy questions. If we make the colonies sick with chalk brood or American foul brood, do they increase resin collection? Do they send out more foragers to collect resin? And so to study this, we set up a bunch of small 5-frame nucleus colonies on these stands, and then we would close the colony up for 15 minutes, and we'd count the number of foraging bees returning with either pollen or resin. And I'll tell you that if you do that, for every 100 pollen foragers you see, you'll see maybe 1 or 2 resin foragers. They're just very, very rare. But when we would count the number of resin foragers for 2 weeks, and then we would make the colony sick, and then we would sit in front of them again and count the number of resin foragers again for another 2 weeks. And every time we did this, and this is just 1 year's worth of data from 2014, but every time we did this, every time we challenged them with chalk brood, Ascosphaera apis here in gray, they did send out more foragers for resin. We saw more bees returning with resin on their back legs. And if we challenged them with bacterial pathogen, Penibacillus larvae, they increased a little bit, but it wasn't different, statistically different, than the control bees. So it's very, very curious to us. We don't have an explanation for it, but when you make bees sick with a fungal pathogen, they send out more, they collect more resin and they send out more resin foragers, but not with a bacterial pathogen. So again, we will try to explore why that might be. We're not the only lab doing research on propolis. There's some folks in Germany, and Sarah Leonhardt did some of this work in Brazil, where they found that if there's a lot of natural propolis in a hive, especially the level of viruses in the colony did not increase as much as colonies that did not have as much propolis. The effect on Varroa destructor, Varroa mites, was not as severe. It didn't seem to have that much effect, but the viruses, it did seem to slow down their increase. And also in Italy, they found that when colonies had a lot of Varroa, they sent out more resin foragers using kind of the same methods that I just showed you for chalk brood and Penibacillus for American foul brood. So where are they getting these resins anyway? And so this will be for our area, and I don't know where you all are from. It will depend on your area, but this is Mike Wilson in this nice looking bee beard. And this is all of these, if you go back through this talk sometime and you look at these references, these are all the published works that we've done. So what Mike did was he collaborated with some plant chemists, and he used reverse phase liquid chromatography followed by mass spectrometry, kind of complicated, but what it does is break down the resins into a fingerprint. And he would go and collect resins from different trees, and he would take resins off the hind legs of bees when they were returning to the nest. So you grab a bee, you chill her on ice, you very, very gently remove her pollen load into a vial, you take it into the laboratory and you do this chemical test. So there's yellow and red propolis and white sometimes. And we sent him all around our University of Minnesota campus, and he climbed 5 pine trees, 3 spruce, 3 fir, all these trees, and collected resin from them. And from that, and then we collected what was on the bees' legs. And from that, we found that in our area, bees are predominantly collecting propolis from cottonwood, Propolis deltoides. They collected a little bit from balsam poplar, but you see Populus and Populus, most of the bees, and this is true in Europe also, are collecting resin from cottonwood trees or balsam poplar or hybrid poplar or aspen, also in the same genus. So we looked at those resins and we ran some tests to see what their antimicrobial properties are. And you don't need to understand all this, but they're listed in order. These plants are listed in order of the highest antimicrobial activity to the lowest, but these are still pretty good. And here's where cottonwood resin falls. But these 3, white spruce and other conifers, pine, some more pine, here's balsam poplar, they have higher antimicrobial activity, and yet the bees are collecting mostly cottonwood. It's very curious. So we don't think these are toxic to them, but I think this is just more prevalent. We're not really sure. These are still unanswered questions. We're trying to understand why they would just collect more Propolis deltoides and not the really strong medicines, if you will, from white spruce and other pine trees. So all of this tells us that the propolis envelope is really good for bees. It provides constitutive or everyday benefits to bees' immune system. It helps them reduce their disease load. They collect more resin when they're infected with fungal pathogens, chalk brood, but not with American foul brood, bacterial pathogen. Kind of curious finding. Bees in our area, the upper Midwest, collect resin mostly from cottonwood leaf buds. And we did find some cottonwood from Nevada, a different species of cottonwood that had unique antibiotic properties that could be used in human medicine because they were unique antibiotics completely. So coming back to our Langstroth beekeeping equipment, our modern day equipment, this again on the right is a beehive, a tree cavity just stuffed full with propolis. And this is what you see in our boxes. The bees put it in where we hang the frames and the frame rests and between the boxes and a little bit here and there in the box, but they never line the whole inside of the box because the wood is very smooth, I think. So how can we stimulate our bees to create a natural propolis envelope? And what you're looking at here is a hive body that's been roughed up. It's just very rough lumber. And Brushy Mountain actually is working with us now. They sent us some boxes that we're testing this summer that had very rough lumber. And we have 10 colonies, maybe 8 colonies that are in this equipment with very rough lumber. And we're comparing the immune system of bees that are in this kind of colony to immune system of bees that have the propolis traps in them. So this is Maggie Shanahan, a brand new graduate student, and she's comparing bees put in colonies with the propolis traps, like Renata ran her experiments. Here's the roughed up colonies, brand new, hadn't put bees in them yet, from Brushy Mountain. And a different beekeeping company sent us boxes that had very thin 16th-inch striations in them. And we also put colonies in these kind of boxes. And I'll tell you right now, the jury is still out. It's still, only mid-July in Minnesota. There's a little more propolis in the colonies with the propolis trap than there is in the rough boxes, but the rough boxes have more than this type of striation. So stay tuned. We're trying to figure out the best way to help beekeepers help their bees make a propolis envelope in their own colonies on their own. And I'm going to finish with talking about a new bee breeding program that we're starting. Again, I'm working with Mike Simone Finstrom, who I said is now working at the USDA lab in Baton Rouge, and my technician, Gary Reuter. And what we're doing is selecting bees that produce a lot of honey, that survive our winter, because we have very severe winters here, that build up very well in the spring. They're gentle, and they bring in a lot of propolis. So this actually is a colony from Brazil, and because in that area the bees bring in a lot of propolis, and beekeepers are able to remove slats of wood from the side of the box one by one, and the bees just build this huge wall, very thick wall of propolis. Our bees don't do that, but I would love to be able to select bees from among our European-derived bees that bring in this amount of propolis. So we want them to be propolis collectors. I'll come back to that. We want them to be hygienic, and we want them to have low mite population growth. And so the way we're doing that to see how much propolis they collect, we've taken these propolis traps and we've glued an acetate sheet onto them. And I'm sorry, this photo's cut off a little bit, but this propolis trap, we cut it so that it just hangs in the frame rest like a frame. And we put it on either side, the long sides of the box, and we also cut a short one for the front part of the box that we stapled on. And the acetate sheet we can see through, and the propolis doesn't stick to the box. It allows us to remove it and look. So we're finding colonies that are bringing in the most propolis in a 2-week or a 3-week period of time, and that are hygienic. And we're using the freeze-killed brood test where we pour liquid nitrogen into a tube, and colonies that remove the most freeze-killed brood within 24 hours. They are going to show resistance to chalk brood and American foul brood, and some resistance to varroa. So we want propolis collectors, good hygienic bees, and also bees where the mites are not growing very rapidly in colonies over the season. So for that, we take samples of mites on adult bees and we do an alcohol wash. In this case, we could do powdered sugar, but we're scientists, so we use alcohol wash. And we also sit down and open up 200 cells of sealed brood, and we go through them one by one to see how many of the pupae have a mite on them. And so then we get a % infestation of mites on adult bees and mites in the brood. We do that in the early part of the season, and we'll do it later in the season. So we're picking colonies that have low mite population growth, very hygienic, and good propolis collectors. And I'll be raising queens from these colonies and inseminating them, and we'll start a new line of bees. But stay tuned on that one. So I'm going to stop there and let people ask questions. And thank you very much for your attention. And I hope if I wasn't clear in any point that you can ask questions and I can clarify it. Thank you.
And thank you so much. That was a lot of great answers to those initial questions that you guys had. You know, I'm sure your research team is always developing these new questions as you find answers. So we'll stay tuned as you continue to progress down the propolis and the health of the colony. And it's also exciting to see a potential new breed come from this. So if you ever need somebody to test out that new breed, I'm more than willing to. Just send me a call to North Carolina. So again, just out to the listening audience, this is recorded, so you can go back, you can view this on our YouTube channel, and we are opened up to questions, and there's a lot of coming in. So I'm just going to pull these out to Marley here, and we'll go ahead and start off. Would it behoove us to paint the interior of new boxes with a dissolved propolis solution? I'm assuming like Mike did in the initial research. So would it help beekeepers to go through that process of coating the inside of the hive bodies with this propolis solution?
Yes, it would, but it's kind of a lot of work. So it means you need to let the bees bring in propolis, then you need to harvest it, then extract it. So that's why we went to stapling the propolis traps in the inside. So we just wanted the bees to do it themselves so that we didn't have to take it from them and then give it back to them. But the short answer is yes, it would help if you have access to a lot of propolis.
Okay. Do you know if resin bees line their nest with propolis?
I don't know what they mean by resin bees. Bees in the wild, feral colonies, wild colonies that nest in trees, they all line their nest with propolis.
Okay. And it sounds like
I'm sorry, I'm talking about honeybees in that case, but maybe the question resin bees was a different species of bee. I'm not sure.
well, somebody had asked about wild native bees. Do native bees, presumably mason bees, bumblebees, more of the native bees collect propolis as well?
No, there are stingless bees in the tropics that bring in a lot of resin. And there's one or two species of native bees, some of the stem nesting bees, Hylaeus, that are resin collectors, but they're not very, these bees are not very common, and they don't, so they're just lining the stem of plant material with a little bit of resin.
And you may not know this, but this is a question. What are the benefits of propolis for humans? A lot of people collect propolis. So what's the benefits for humans?
Sure. Well, there's been some research on the benefits for humans. Most of it's been on oral, your mouth infections, so gum disease or oral herpes. If you have any kind of infection in your mouth, if you just take some propolis tincture and put it in, it's very, very effective. It helps some people with wounds. If you have a wound anywhere on your body, it will stain your skin, of course. But otherwise, some people take it when they have colds or sore throats. They'll put some in tea. And that's all I really know. I think there is some research in Japan on any benefits for tumors, but I don't know if it is effective for cancer.
Okay. That's more than I knew. How long does propolis retain the beneficial properties?
Right. So bees will be collecting propolis as long as the growing season is. So as long as they'll just bring in our area, cottonwood resin from, May, and as soon as the leaves are out, all through September. But through the winter, the propolis does lose some of its antimicrobial properties. But then when bees start foraging again, they just keep stuffing the new resin back in, and it regains, the new stuff. They just stick it on the old stuff, and so they just have layers and layers of it, and it regains the properties again.
So kind of a follow-up to that, this is me personally asking this. So I have a hive. I removed out the brood box as I condensed it down for winter. And that brood box, usually I'll either soak it in a bleach solution, or I may come back in and burn the interior wall to kill off anything in there. Is that something I should be doing if they have a lot of built-up propolis, or should I retain that propolis build-up and use it in the future year?
Well, you can retain it. Mostly if it's in your way, you're going to need to remove some of it. So that's why we're working so hard to figure out how to allow the bees to make the propolis envelope. And when they do that, it's not so much in our way. They're not putting so much between the boxes and in the frame rests when they're allowed to make the envelope. But mostly, the propolis is really good for them. So as much as possible, leave it in there.
Okay. So this is multiple questions. So I'll sum it up in part A and part B. So part A, I know you have a lot of great research published out there. The plants that you listed out, trees, plants, I know that there was some conifers on there as well as some populus. Do you have that list of plants or trees published somewhere? And a follow-up B, what should people be looking for in their natural habitat that would indicate propolis producing plants?
Well, that list, it's in the publication. I mean, I can scroll back in my slides to this publication at the bottom of this slide, if you can see it. And if you go to our website, which is blab.umn.edu, and you go to the research and publications page, you can see all of our publications. So yes, that's all published. But basically, you're just going to look and see if your bees are bringing in resin, and you just have to sit in front of your colony and look at them. Sorry, I'm leafing through these slides. And see if they are bringing in resin, or you can just see it if they deposit on the walls. And it's not so important to know where they're collecting it from, because all of it is really good for them as far as we know.
Sorry, again, there's a lot of great questions out there, and I may not get to all of them. I apologize. Marla, if people have future questions, can they reach out to a specific email address or some way to reach out to you to get these answers?
Sure. Like I said, if you go to our website, so I'm going to scroll all the way back to the very first slide here. This is our website, blab.umn.edu, and you go through there and you can find all of our references, and there's some that are scientific and some that are more for the general public. And also on the website, you'll find my email address. I must be very easy to reach because I get a lot of questions. So I'm happy to answer if I can.
Okay. Well, you finished up with your new potential breed of queen. There were questions about Caucasian bees. Somebody mentioned African bees. Is there a specific line of or breed out there currently that would help in this encouraged propolis building?
Well, I've heard that Caucasian subspecies do bring in a lot of propolis, and Sue Kobe and Steve Shepard from Washington State are introducing a few Caucasian, well, they're introducing the semen from Caucasian bees and doing crosses. So I hope to try out those Caucasian bees, but mostly I really don't, there are colonies within any race of bees that collect a lot of propolis, whether they're Italian or Carniolan or the Russian breed, or there are colonies that bring in a lot of propolis, and it's a genetic trait, and I'm just looking for them no matter what genetic background they have. So I just want them to be propolis collectors and hygienic and have slow mite growth, and otherwise I don't really care where they come from.
Well, thank you so much for all this information. It was tremendous. I learned a lot from it. I know that our listening audience learned a lot from it. It is a recorded session, so you can go back in and review all this information that was given. Also, please go to B-Lab at the University of Minnesota. Look at all the research that has been done. I know that propolis has been a focal point, but they've got a lot of other great sources out there that can help people become better beekeepers. So please check them out. Look into this. Marla, again, thank you so much for everything that you've given us. It's a lot of great information, and I am concluding the webinar. Thank you, everybody, for listening in.
All right, thank you.
Bye.